Welcome to our next topic. Today, we will be understanding biofilm formation. So as you can see, I have a couple of structures on this side, and using those structures, I will be explaining biofilm formation in a very, very simplified way, but at the same time, I'll also provide detailed information that is going to be very, very useful for your understanding of this particular topic as far as the exam is also concerned, right? So I'm taking the model organism called Pseudomonas aeruginosa. And if you are not aware of that organism, let me also briefly explain to you what Pseudomonas aeruginosa is. Right over here, what I'm showing you is a bacterial cell, right? So when we talk about biofilm formation, so biofilms, they are structured communities created by these tiny organisms. So organisms, these are, the size of the bacterial cell is very, very small. You cannot see this bacteria with the naked eye, right? So what you need is an instrument called microscope. Under microscope, you can visualize these tiny organisms, and these are present everywhere, basically. They're very, very small. You can't see them. But when they are present in communities, let's say humans are present in villages, right? Cities. So these cities can be visualized from space even. Same thing, bacteria, they also form communities, and those bacterial communities are called biofilms. A lot of research is going on in and around a biofilm, that how bacteria, they are, very, very tiny organisms, very simple organisms, are able to create these complex communities. And what happens inside those communities, what happens inside those larger structures that are there in biofilms. So a lot of research, including my research, is trying to understand is what is bacterial biofilm, how we can understand the formation of biofilm, how you can basically destroy the biofilms of the bacteria that is not good for humans, right? So these are some of the things that we'll be focusing, and I've divided this lecture into multiple parts. So I want you to cover all of them, because as you can see over here, biofilm formation is not just a single stage; it's a multiple stage. You have attachment, you have colony formation, you have maturation, you have dispersal. All those things are there. So we'll be understanding all those things step by step, right? Okay, we'll start with the step one, which is surface attachment. But before that, let me give you a little bit background on Pseudomonas aeruginosa biofilm formation, right? So Pseudomonas aeruginosa is the bacteria that is responsible for nosocomial infections, also known as hospital-acquired infections. So when patient goes to hospital, Pseudomonas aeruginosa can colonize those patients and cause infections, right? That's why they are known as opportunistic pathogens, because they survive on those individuals who are already having low immunity. So next is biofilms, right? So what are biofilms? Pseudomonas aeruginosa is one of the organisms which is known to create very, very complex biofilms. And biofilms, they are structured microbial communities, and which are attached to the surface, and which is also embedded with a self-produced extracellular polymeric substances, which is also known as EPS matrix. I know it's too much, right? So let me break it down for you. So EPS is nothing but the glue that will try to fix bacteria together, and because of that, it can create larger structures. So EPS, full form of EPS, is extracellular polymeric substance. So you can understand that it is something extra apart from bacteria, right? Anything that is extra other than bacteria is EPS. Next is in Pseudomonas aeruginosa, biofilm formation is highly regulated process, and because of that, bacteria is able to resist antibiotics. It can evade host immune response also. That is one of the important part of the understanding of biofilms. Next, we have different stages of biofilm formation. You have initial attachment stage, you have irreversible attachment stage. Third, you have microcolony formation, and then biofilm maturation. After that, once it's matured, further development and dispersal. So you have roughly divided biofilm development into, we have roughly divided biofilm development into these individual stages that we will be understanding. In this particular video, I'll be showing you a little bit about initial attachment, which will include reversible as well as irreversible attachment. Reversible attachment is also known as initial attachment, right? Okay, so let's use our amazing illustrations, right? So that we can understand this process much, much better. At the same time, I'll also show you that what are the factors responsible for this attachment. All right, as you can see over here, I have this bacteria that I've created, And bacteria, I'm sure that you have seen my previous videos also on bacteria, bacterial cell wall, bacterial surface structures and all. So now you have pretty good idea about bacteria, but I'll introduce a little bit on that. So bacteria will have surface structures. It will have membrane, cell wall. It will also have locomotor structures. Many of the bacteria that are not able to move from one place to another, they do not have locomotor structures like flagella, right? They also have surface structures like these tiny structures. I hope you can see them. They are pili or fimbriae. They are able to help bacteria to attach to the surface, right? And I'm going to show you that process, that docking process with the surface. Okay, for that we need the surface. Let me decrease the size of this. Okay, so this is the bacteria that we have, right? And then bacteria, let's understand this. Bacteria is trying to attach to the surface. So right over here we have the surface. Let me copy it. And surface means it can be anything. I'm just creating a solid block over here. Surface, it can be anything. It can be abiotic surface, it can be biotic surface, right? And usually abiotic means non-living, biotic means living, so they can also form biofilms on human organs also, right? So what happens is, let's imagine the bacteria is motile and it's floating around like this. So bacteria is moving in the liquid environment, and when bacteria is moving in the liquid environment, what you have is these locomotor structures like flagella. They are working very, very hard. This structure that you can see, the tail of the bacteria is working very, very hard, and bacteria is able to swim through the liquid environment very, very effectively, right? So it can swim through and it can move towards the nutrition. It can move away from the environment, which is it's difficult for this bacteria to survive. So here the flagella is playing very, very important role because it's a locomotor structure, so it can swim around, right? Next is when bacteria bacteria reaches to the environment where you have a lot of nutrients, so bacteria will decide to settle down. So for settling down, bacteria will basically sense the surface, and it will have receptors to actually help the bacterial cell to attach to those receptors, right? So as you can see over here, let me also increase the size of this so that it's much more visible. All right, so as you can see over here, bacteria has these tiny structures. We call them pili fimbriae structures. They help the bacteria to actually, slowly dock to this particular surface like this. And now you can see it got docked, right? Those tiny structures, they now are in place. They have attached to the surface, and this is where we call it as, irreversible attachment. When these pili fimbriae structures, they are fixed on the surface. But there is another stage, which probably I should have explained earlier, where these structures are not playing important role. Bacteria just roaming around like this, right? Sensing the surface. And then you have some of the interactions which is not associated with these structures is happening. That is the initial attachment. So it will go and, have the interaction with the surface, trying to map down. And at this particular stage is the irreversible stage. Here, there is no structures that are involved in strong binding with the surface. So you have this bacteria just exploring the surface. So that is the initial attachment, right? And it can be irreversible. It can be reversible attachment. But when it comes to attachment with these structures like pili, fimbriae, and bacteria is just docking on the surface of it, and that is probably the irreversible stage where bacteria is fixed. Now it's not going to move anywhere. Okay, and what bacteria does after this, it's going to reduce the function of these locomotor structures. Now it's not going to move around too much, so they're going to slow down. Probably all the genes associated with this bacteria, they're going to slow down, and bacteria has acquired the stage called initial attachment as well as the reversible attachment stage, right? So it's very, very important for us to understand that there are two stages. One is initial attachment stage. The second one is irreversible attachment stage in case of bacteria. All right, so now let's formally understand the initial attachment process and see what are the components that are involved in the attachment process. The first thing that I would like to mention over here is cells they're going to approach using flagella-mediated swimming motility. Whenever there's the motility mechanism of the bacteria which is induced by flagella, it's known as swimming motility. Next one, the stage which is we have already discussed is a reversible attachment, and that is involving the interactions like hydrophobic interactions with the surface, van der Waals forces, and third one, electrostatic electrostatic interactions. These are the three important interactions that are involved in reversible attachment. Okay. The next one is surface sensing mechanisms. So surface sensing will start from here, where we have already discussed that pili structures, as well as, flagella also plays very important role. You have fleece protein of flagellum that enables swimming motility and initial contact with the surface. Structures like type 4 pili, which is pil A, pil B, and pil T. These are the proteins that are important in case of motility, which is called twitching motility. We'll understand twitching motility later on in this particular topic. Next is pil Y1 protein. So, in type 4 pili, you have various proteins, but pil Y1 protein, it's a mechanosensor. It's a mechanosensor which can detect the surface contact. We have worked in very much detail in our research publications on pil Y1 protein, and we try to understand and how it interacts with the surface and how we can block that interaction. Next is fimbriae and adhesins. There are other proteins that are present on surface. They can help the bacteria facilitate that particular interaction with the surface, right? So next is you have, there are so many other things. At this particular point, bacteria also start producing EPS, and that EPS production with the attachment with the surface can lead to further strengthen will cause it to become permanently fixed to the bacterial surface. At initial stages, when these particular components are working, what happens is the bacteria might be able to, detach from the surface. But when you have EPS production, when you have fimbriae, when you have pili, all those structures working together and they are able to fix the bacteria with the surface, then it becomes irreversible stage. But we will understand these polysaccharides, these extracellular matrix later on in our videos. So I'm not covering them right over here, but you need to understand the difference between the reversible and irreversible stage, right? Reversible, when the bacteria is sensing the surface, it is trying to explore the surface using those surface structures, including pili, flagella. It's not fixed over there, right? But when bacteria is fixed with these structures, along with some of the extra polymeric substances that are getting secreted, the bacteria becomes irreversibly attached to the surface. So that's the second point. But usually you have this stage called attachment stage for all of these things, right? Okay, so now from here we will move on to the next stage, in detail understanding that what are those molecules that are involved in bacterial biofilm formation, such as microcolony formation. All right, so let me show you that initial attachment or surface attachment illustration that I've created. So I have created this illustration, and let me make it a little bit bigger. I hope you can see this, right? Let's ungroup these individual components so that I can move them around. So as you can see, slowly bacteria, they will gather and they will come very close to the surface, and you can see one structure over here that is slowly sensing the surface, and now it is getting attached. So now the attachment is with those surface structures, both adhesins, with the adhesins and also with type 4 pili as well as flagella. I can move it a little bit like this. Okay, so interaction is happening with all those structures. Now bacteria has also started to secrete those extra polymeric substances, and now bacteria is firmly fixed with the surface. All these bacteria, they are just exploring the surface. As you can see over here, this bacteria is moving very close to the surface. Now there is a little bit of interaction, not permanent interaction, but it's irreversible. It's reversible, right? But slowly what they do is they sense that bacteria is getting settled down. Other bacteria, they also come and fix to the surface like this. Slowly they will have that interaction with their surface structures, and you can see the bacteria, they are slowly getting attached to each other, also to the surface, leading to further structures which are also known as microcolonies. And we'll understand microcolonies because I have that structure over here. So that's the second part. Okay, I hope you understood all those points regarding the initial attachment as well as the irreversible attachment. Now let's move on to the second one, which is microcolony formation or colony formation. And thank you for watching this video. If you have liked this video, please give a thumbs up, because that helps us create these kind of contents in more frequently. Another thing is I've also created a resource called Research Goal. Research Goal is a website where I host a lot of courses that are going to help you in your research. especially skill-based courses are there, right over here in this channel. I provide information basically on the topics, theoretical aspect of the topic, but practical and skill-based learning is there on Research Goal. I'll put the link in the description box. Please go and check it out. I'm sure you're going to like a lot of the courses that are really important. There are courses that will help you navigate the PhD process. There are courses that will help you in writing reviews. There are courses that will help you bioinformatics, computational drug discovery. So many things are there. Please check them out. Okay. Thank you so much.